Before we go on, we'll have a quick review of how the mass spectrometer generates data. The MS continuously scans a mass range, typically 350 to 1500 Daltons when using electrospray, and it does this for about half a second per scan. If interfaced with chromatography, sometimes there are no ions in the scan of the spectra. But depending on the complexity of the peptide digest injected onto the column, there could be 1 ion or 20 to 30 ions in a scan. In the case of this example, we've zoomed in on a small part of the mass range, and there are likely other ions present if we zoomed out. In the nanoseconds after every scan, the computer examines the spectra and looks for multiply charged ions such as this one highlighted. The instrument then uses quadrupole 1 to select the C12 isotope and exclude any other ions being sprayed in. The 785.8 Dalton ion is accelerated into the collision cell where it collides with nitrogen gas molecules and undergoes fragmentation, with different molecules of the same peptide being broken at different places. The fragments are then collected and pulsed into the time of flight detector for measurement. The result is a spectrum like this, known as an MSMS or a fragmentation spectra. The original unfragmented ion, or the parent ion, is indicated on the scan, and some of that parent ion has managed to escape being fragmented. The other major peaks in the spectra are reflective of the amino acid sequence of the peptide, and the difference in mass between peaks will be equal to the mass of an amino acid. So how do we go from this unlabeled spectra to this annotated spectra? First, we need to look at the nomenclature, or the language used to refer to fragments of a peptide. When a molecule of a peptide gets broken during collision-induced dissociation, it usually only forms 2 fragments. Different molecules of the same peptide can get fragmented in different places in the chain, and thus we produce a ladder of fragments. The fragments will be singly charged. So the MSMS spectra can have fragments representing the peptide sequence from the N-terminal end and have fragments representing the sequence from the C-terminal end. As both series are in the spectra, things can get a little complicated. The shorthand way of assigning peaks in a spectra to an amino acid bond is to define the B and the Y ions. If the peptide molecule is broken through the peptide bond, the N-terminal fragment is called the B ion, and the C-terminal fragment is called the Y ion. It should be noted that the numbering occurs in opposite directions. For tryptic peptides, the Y1 ion, or the C-terminal amino acid, will be an arginine or a lysine. So we end up with a series of fragments that will hopefully reflect the entire amino acid sequence of the peptide. This will ultimately depend on the amount of peptide present and the sequence, as certain amino acids can cause preferential fragmentation through a particular bond, and certain ions will be more intense, which explains why the peak in our spectra are all different heights. There are some general rules that we can follow when trying to sequence a peptide from an MSMS spectra. First, a B ion's mass will be the sum of the amino acids in its sequence plus 1, the mass of a hydrogen ion. The N-terminal, or B1 ion, will be somewhere between 57 and 186 Daltons, the mass of the lightest amino acid glycine and the heaviest amino acid tryptophan. It will be the amino acid mass plus 1. The largest B ion should be found somewhere between 57 and 186 below the mass of the intact peptide. Because of the way atoms rearrange after fragmentation, the mass will be the parent's mass minus the sum of the mass of the amino acid plus 18, the mass of water, which is the same as the parent mass minus the mass of Y1. Starting from the other end, a Y ion's mass will be the sum of all the amino acids in the sequence plus 19 because of a hydroxyl group and 2 other hydrogens. If there are any modifications to the N or C terminal or to any of the side chains of the amino acids, the extra mass must be included in the calculations. Leucine and isoleucine are the same chemical composition and thus the same mass and can't be distinguished. And finally, if the protein was cut with trypsin, then Y1 should be either lysine or arginine. So now we're going to work our way through an example developed by Karen Jonscher and published by Proteome Software, where you can find other resources about MS/MS sequencing and database searching. This spectra comes from a quadrupole ion trap. The mass of the precursor was 1449.38 Daltons, and it was doubly charged. We'll start by looking for peaks that are below the mass of this precursor ion. So we're going to look for possible Y ions. That means the length of the peptide minus the first amino acid between 57 and 186 mass units below the precursor mass. So here's our spectrum, and we have 1449.38 as our precursor, as shown in the top right hand corner, and our highest mass ion is 1337.5. If we subtract that from 1449, we get 113, which is either leucine or isoleucine. So the N-terminal amino acid must be leucine or isoleucine. We will find complementary ions further on through the sequence, further on through this example. The next ion down is 1276.5. Now, the distance between 1337 and 1276 is too close to be the next amino acid from the N-terminal, but if we subtract 1276 from 1450 and take into account a water molecule, we get 156, which is the mass of arginine. Now we know we digested with trypsin, so it is most likely that this ion is the largest B ion in the sequence. Next we look down at 1238. Again, this is too close to 1276, so it must be the next one in the sequence after 1337, starting from the N-terminal end. What these orange indicated fragments are is the amino acids firstly losing the first amino acid and the charge being carried on the C-terminal end, and the second one, 1238, is the peptide missing the first two amino acids and the charge being carried on the C-terminal end. So the difference between 1337 and 1238 is 99, which is the mass of valine. So we say that the next ion in the sequence from the N-terminal towards the C-terminal is valine. We keep going in this manner, and we can assign amino acids to different fragments as we go through. And you can see that going from the orange, which is going from the N-terminal to the C-terminal, we now have leucine, two valines, and a phenylalanine. And then the purple ions, which indicate that we have an arginine, a glycine, and then either a lysine or a glutamic acid. Now, because we cut this with trypsin, it's most likely to be the glutamic acid, because if it was lysine, it would have cut that last part of the peptide off. So now we need to verify our assignments. So now we start to look for complementary ions down in the low mass range of the spectra. Now in ion trap data, we very rarely see the B1 ion. In quad-TOF data or in Orbitrap data you would, but in this case we won't. So we're going to start looking for the mass of B2, which will be the mass of leucine or isoleucine plus the mass of valine. If we add these two ions together, we get 213 once we account for the hydrogen atom, and lo and behold, we have an ion at 213 in our spectra. This confirms the leucine-valine part from the N-terminal. If we then look for leucine-valine-valine, we end up with 312. There it is, 311.8, it's pretty close. Next we look for the complementary phenylalanine. So if we add 147 to 312, we get 459, and there is the ion there. In this way we can start to look through all of the ions. So if we went back and added together the arginine plus glycine starting from the C-terminal, we would end up with the mass for Y2, which is indicated by the orange bar. If we added the mass of the Q, we'd end up with Y3, and this is also indicated. So in this way, you can build up a complete pattern. What you'll notice, and the most important thing to notice here, is that all of the major peaks are explained. All of the major fragments should be labeled and explainable by the peptide sequence, and if they're not, something has been calculated wrong, or there is some kind of a modification or loss that hasn't been accounted for. For instance, things like serine and threonine can lose water, and therefore you would find a major peak at 18 Daltons less than what you would expect. And these are the masses of the amino acids that we can use to determine our sequence. In the next lecture we're going to discuss how to do this in an automated fashion, because to do this by a calculator for thousands upon thousands of spectra is just completely impractical.